Okay, well, good morning, everyone. Thank you for coming out to hear this. I was asked by the organizers to talk about the biodiversity issue from the perspective of someone who works with microbes. And what I'm going to try to do this morning is to provide an overview of the work of many people who have been looking at the relations between biodiversity and ecosystem functioning, mostly in microbial systems, some in the laboratory, some in the field, and to try to focus on some of the major issues that have cropped up and to provide an overview of some of the important patterns that have emerged from these studies. So as Judd pointed out, there's a lot of interest in this particular topic. And in particular, if we look at this side of the graph over here, if you're concerned about losses in biodiversity moving in this direction along this axis, you might expect that at least in some systems you could either see a corresponding decrease in some functioning if species are contributing in unique ways to that particular function, or if you have a lot of redundancy in systems, you might in fact be able to lose lots of species, maybe almost all the species, before you see some sort of change in functioning. And that's the crux of part of the debate. What kinds of patterns do you see when you look at different kinds of systems? The other disagreement that's occurred is whether in fact diversity is a cause of function or whether in fact diversity is a consequence of other aspects of the ecosystem, such as this productivity over here. And what I'm going to suggest is that these are not mutually exclusive possibilities, and in fact there's plenty of evidence for both of these things, and we shouldn't see this as either or. The answer to this question, biodiversity cause or effect, is in fact yes. They're both important under different kinds of circumstances. What I'm going to do next is spend a little bit of time talking about why I feel that microbes are very important model systems for looking at these kinds of issues, and then from there go through a whirlwind tour of where you find different kinds of patterns showing up in the studies that have been done. Okay, so why use microbes? They're wonderful models, and in much of what I'm going to talk about today, I'm not trying to explain patterns in natural systems. Rather, I'm going to be telling you about studies that have been using microbes as models to manipulate diversity over a range of different trophic levels. And that's one of the great things about microbes. You can build food chains, you can build somewhat more complicated food webs, you can vary the diversity within the different functional groups in these systems, as any number of people have done here. And by doing that, you can understand how diversity distributed across an entire model food web is likely to influence aspects of functioning. That's something very different from what's been done in most terrestrial studies of biodiversity that have focused largely on plants. And as you'll see, the places where diversity varies can be very important in influencing the outcomes that you see. The other important thing I want to stress with this probably overly busy graph is that by using microbes, we can get around one of the problems that occurs when you do a diversity experiment with slow-growing organisms. And again, the example of a slow-growing organism is a terrestrial plant up here, where what you put into a diversity experiment at the outset very much determines the outcome. Densities don't change very much. And there's been a great deal of argument and discussion as to how one should best set up these kinds of studies. The beauty of microbes is that all you do is build a community or a food web by inoculating things at very low initial densities, and then they grow up according to the kinds of interactions that they have in their system, and you end up with a system where things are not evenly distributed across species. There are common things, there are rare things, and it's much more like the patterns of dominance and diversity that you're likely to see in a natural community. It also means that when you see diversity effects, it's mostly due to these changes in dynamics over time, as opposed to just changes in the somatic growth of organisms, and that's a particular advantage for getting at some of these questions. Now, that said, microbes are not perfect. They offer some drawbacks as well. I should also point out that during today's talk, I have a very ecumenical view of microbes. I'm talking about bacteria, I'm talking about protists, I'm talking about phytoplankton, anything that's small enough so that you need a microscope to appreciate it. The problems are these. When we do these studies, we're mostly limited to studies of culturable species, and that means we can only do these studies with maybe 1 to 5% of the organisms that we know about, which means we're leaving a lot out. And that's potentially a big issue. The other very vexing thing is that it's very difficult to do these experiments under natural situations. Microbes don't stay in cages as much as you would like them to. And that means that you can't do the kind of field manipulations that people who work with larger organisms can readily do. And the other thing that's related to this culturability problem is that oftentimes we can only use a very small fraction of what you would ordinarily find as the natural level of microbial diversity. That's not an issue if you're using these systems as a model, which is what many of us do. It is an issue if you're trying to use those models to explain the full range of processes in natural microbial systems. So that's something to keep in mind. To make that point a little more clear, on this side of the graph you can see the range of species richness that's been used in a number of different studies, some older, some much more recent. Tom Bell and his colleagues currently hold the record for manipulating diversity from 1 to 72 bacterial species, which sounds pretty good until you look at what the probable natural range of bacterial species richness is in natural communities. At least in terrestrial communities, it could be somewhere up in the millions. In aquatic communities, it's probably more in the thousands or tens of thousands. And the point is that microbial experiments done in microcosms in the lab might be finding responses to diversity over this range that simply don't apply to what we would find in natural systems. And again, that's only an issue if you're trying to use these systems to understand natural patterns. If you're using them as a model, which is what many of us do, then this is still a perfectly valid way of looking at these kinds of patterns. Another aspect of the controversy is whether it even makes sense to talk about variation in microbial diversity. And this has more to do with an ongoing controversy about the extent to which microbes actually vary in diversity over space and time. And there are two very different sets of opinions here. The first is typified by this quote from Bland Finlay, which basically suggests that microbes disperse so widely and are so abundant that they're everywhere, and that it doesn't really make sense to talk about variation in their diversity because there isn't much. And that's one point of view. There are other points of view, of course. Willem Feusner, who's an excellent protist taxonomist, has suggested that, in fact, there is a lot of biogeographical variation in the distribution of at least some protists, and that means that it should be interesting to look for variation in functioning along that. Similarly, Jennifer Martini and her colleagues in a more recent review suggested again that if you look carefully at particular species, you'll find that there is variation in their distribution. And about all we can say at this point is that this is an ongoing disagreement. And we have this disagreement because our knowledge of the natural distribution of microbes in most systems is so poor that we can't really say to what extent things are either universally cosmopolitan or display a high degree of endemism. And I'll return to that point at the very end of this talk when I talk about some of the challenges that we face. Okay, so what are the patterns? What I'm going to do now is do a very quick tour through some of the major patterns that I think have emerged from studies of biodiversity in microbial systems. These come from a range of laboratory and field studies, and they address a number of different things. First, we're going to look a little bit at those productivity biodiversity patterns, and then look at some aspects of functioning, stability and reliability, end up a little bit with some work that's been done with invasibility and also some interesting indirect effects that have emerged from some of these studies. So here we go. If you do very simple kinds of experiments in the laboratory, in this case these are some results from Rhys Casson and his colleagues, one of the things that you can see is that you can recover, in fact, these kinds of relationships between, in this case, productivity and diversity in a very simple laboratory system where you have a single species of Pseudomonas that's allowed to diversify into different morphs. Pseudomonas fluorescens does this with great rapidity. And the only thing you need to generate this kind of curvilinear relationship between biodiversity on this axis and productivity is a stable, structured environment that allows these species to basically break up a test tube that they're cultured into three different kinds of niches. You don't need predation, you don't need disturbance. All you need are differences in resource use to generate that kind of pattern. So you can see these kinds of things emerge in very simple laboratory systems, which again makes this a nice model. But as you're going to hear in this talk and also in Helmut's talk after mine, the world is a complicated place. And in fact, you can do just very slight variations in the way that you set up experiments and generate basically all possible kinds of productivity diversity patterns: flat, increasing, concave down, concave up. In this case, this is a study that was done by Tad Fukami in my lab, and he did this with a set of 16 ciliates that all feed on bacteria. And to generate all these kinds of different biodiversity patterns, all that Tad did differently was to vary the sequence, the order of introduction of the same set of species into the system. And just that added level of complexity is enough to generate this full panoply of different productivity diversity patterns. So it doesn't take much to make things more complicated. The other thing that's worth pointing out is that these are not pure lab artifacts. You can go to natural systems, which is what Irigoyen et al. Did in their study of marine phytoplankton in this case. And if again you look for relationships between productivity and diversity, you can recover again these kinds of curvilinear relationships. Here's the actual data up here, which are a little hard to pick out in this three-dimensional graph. But if you fit a 3D surface to it, what you find is that, again, you get this kind of curvilinear relationship between, in this case, a measure of productivity, and in this case, diversity. So the kinds of things that we see in very simple systems in the laboratory also emerge in much more complicated natural settings, which gives us some solace, I guess. Now, turning the axes around and now looking at effects of diversity on aspects of functioning, and here we're going to be looking at a whole range of different functions, not just productivity, but other kinds of things. I'm going to suggest that, again, depending on the kind of function that you look at and the level or trophic level in which diversity is manipulated, you can find, again, a whole range of different patterns that have emerged. Here again are results from Bell et al. In a lovely paper that came out in Nature a few years ago, where they again manipulated bacteria isolated from a natural system, and here the measure of functioning is respiration. And there's this approximately linear increase in functioning that occurs across the range of diversity that they looked at. This suggests that as you add different species, you're adding unique functional traits in this system, and there's no evidence of this trailing off or saturating whatsoever. And that again is what you might expect for organisms as functionally diverse as bacteria, not too surprising. You can also find negative effects of biodiversity on different kinds of functions, which again weren't really considered in some of the early discussions about the value of diversity. This happens if you manipulate the diversity of consumers and then look at the response of the things that they consume, in this case, algal biomass and bacterial abundance. This is a study that was done by Naeem and Lee more than 10 years ago now. And what they found is that by increasing diversity in that one trophic level above the consumers, you in fact get this kind of decline, which is shown over here with their actual data. And so if you're at the bottom of the food web, increasing diversity at higher levels isn't always necessarily a good thing. You can also get these sort of standard saturating kinetic responses that are consistent with there being some redundancy in the level of functioning that occurs in these systems. Here again, it's a different function. This is decomposition, and this is as a consequence of different richnesses of protozoans that were added to some laboratory systems. This is an intriguing result because the things that are increasing in diversity here are not the things that are actually doing the decomposition. That's the bacteria. The protists are eating those things and manipulating them in other ways, and so this is an indirect effect of diversity higher in the food web, which is changing the way that the microbes that are breaking down carbon sources are operating. And I'll return to this result in a little bit when we talk about some other indirect effects. And you can also get patterns that appear to have very idiosyncratic or perhaps no relationship to diversity. Here again is a different kind of function. In this case, the ability of an invader to enter these systems, where high values mean the invader do well, low values mean it doesn't. And here it's either nonlinear or very idiosyncratic in whatever kinds of effects that the species richness of the extant community have on the ability of an invader to come in. This suggests that this has more to do with the actual composition of the community than it does with the actual richness of species that are there. And then finally, you can have patterns where basically not much seems to happen in response to biodiversity. This is for another measure, cumulative carbon dioxide flux in some laboratory systems. And there's a slight positive trend, but really there's not a whole lot going on when you look at the average. But there is a lot going on when you look at the variation. And that's the next thing I'm going to turn to in talking about biodiversity effects. Diversity can have a large impact on the variability or predictability of these systems. And those results come, again, from a couple of older studies that were published almost back-to-back in Nature a number of years ago from Naeem and Lee and from our lab. And both of these show that if you look at measures of variability, either the standard deviation of bacterial density or the R-square for autotroph biomass, or in this case, the standard deviation of carbon dioxide flux, these systems become less variable and more predictable as you increase the diversity over fairly small ranges of species richness. And that provides another potentially important aspect of biodiversity, and that is more diverse systems are easier to predict in terms of their traits. At least that seems to be the case in laboratory systems. A very recent paper that just came out this year by Tachnik et al. in PNAS also suggests that you can get similar kinds of diversity variability patterns in natural, very complex phytoplankton systems. Here, these are measures of either compositional turnover or variation in resource use efficiency as a function of the number of genera of phytoplankton in these systems. And in both cases, as richness goes up, the variability goes down in these complex natural systems. The flip side of this is also as you increase the productivity in these systems, which is measured here by phosphorus, you see the reverse pattern, which is consistent with a sort of paradox of enrichment. As these systems become more productive, they become more variable. And that's, again, a nice counterpoint to the kinds of things that we've seen in the laboratory studies. It appears to work in natural systems as well. These are, of course, observational results, and what we'd like to be able to say is what happens if you can actually manipulate the system and see how it responds. Are they, in fact, more resilient or more stable? There are ways that we've been able to do this in laboratory systems, where we take a system like this one, which varies over some range, and then at some point perturb it so that we impose a 90% mortality on the organisms in the system, and then simply allow the total biomass in the system to recover over time. That rate of recovery to some reference level gives us a measure of resilience, this kind of thing over here. And then we can see how the resilience of the system to some kind of perturbation will respond to this kind of manipulation. And when we've done that, this is work that Chris Steiner and some of my students did in my lab, what we've been able to show is that at least at some levels, in this case at lower levels of productivity, if we look at a measure of community level resilience, this here as a function of low, medium, and high levels of diversity, which correspond to 1, 2, and 3 species per functional group, you do see a significant increase in the resilience of the system in response to diversity. So not only do you see these things in observational studies, when you actually experimentally manipulate the microbes, you can also see that they have this kind of response. Okay, now I want to finish up with just a couple other more elaborate patterns that have emerged from some of these studies. And one of the things that we've known, in fact, for a long time, again, returns to this topic of these indirect effects of consumers on bacterially mediated processes. A long time ago, Fenchel and Harrison showed that if you look at decomposition of barley hay, in this case, in seawater, one of the things you'll find is that if you just look at what the bacteria do without protozoa, things decompose, but it happens slowly. And if you just add one protist, a choanoflagellate in this case, the amount of decomposition increases substantially. And if you throw in the protozoan kitchen sink, in this case, add a whole mixed group, what you find is that there's an even greater amount of decomposition that happens. And the reasons for this are a bit mysterious, and have only recently been teased out somewhat. Farther along, when we looked again at a pattern similar to this, we found that not only does decomposition sort of depend on the presence of the grazers that are feeding on the bacteria that are doing the decomposition, but having more grazers there leads to an increase in decomposition. And we've delved into that a bit further. There's a very recent study that was done by Dave Gruber and his associates at RUTGERS, and one of the things that Gruber found, which is very intriguing, is if you look at a single species of bacterium feeding on a carbon source, and then add just a single species of protist that's grazing on that, what you find is that adding that protist to this single species of bacterium, again, leads to an increase in decomposition, a decrease in the remaining amount of particulate carbon present in the system. To get that kind of result means that the consumer is changing not bacterial species composition, because there's only one bacterial taxon there, but it's having some sort of effect on bacterial physiology or activity. And that provides one kind of insight into what's going on. Jennifer Crummins, working in my lab, has worked on some similar kinds of things. And she's also found, again, that if you manipulate eukaryotic species richness, you get this increase in decomposition. And that increase in decomposition happens even when the grazers are driving bacterial densities down. Okay, so fewer bacteria are doing more decomposition, which again suggests that there's some either change in bacterial composition or bacterial activity that's being driven by the higher level diversity in this system. So that's one story. Another quick story that I'll tell you about has to do with the relationship between biodiversity and biological invasions. Again, this is an ongoing controversy, especially in terrestrial ecology, where we see two different kinds of patterns. On the small scale, there's a pattern that appears to be related to biotic resistance, whereas if you have more diversity, invaders do not do as well. On larger spatial scales, where typically experiments are not done, but you simply observe the response, you get a completely opposite pattern. As diversity goes up, so do the number of invaders. So what's going on there? Why do you get these different patterns? You can use microbes, again, to tease this kind of thing apart, which is what Lin Zhang did in my laboratory. He manipulated productivity and looked at the effects of increases in productivity on both local diversity in the community and also on how well the invaders do. The quick answer here is that increasing productivity leads to an increase in the abundance of a simulated invader. It also leads to an increase in the abundance of the resident diversity in the community. And when you plot those two things together, you get what appears to be a positive relationship between resident species richness and the performance of the invader. But that result is a complete red herring. It's not driven by local diversity. It's driven by the fact that both of these things are responding to the productivity gradient. And using microbes, we suggest that the reason for this kind of positive relationship is that both of these things are responding to the same causal factor. In this case, it's an external driver of productivity in the system. One final thing that I'll talk about is that you can also use microbes to dissect out the effect of species traits as opposed to the effect of biodiversity in these systems. This is work that was done by Zach Long in my lab, and one of the things that we know is if you look at a trait, in this case cell size, and the abundance of different species in these systems, you tend to recover communities that fall along this -1 line of constant yield. Here's what the experimental data actually look like. We have points below and above that constant yield line. And the thing that determines whether the points fall below the line or above the line is the biodiversity in the system. Less diverse systems tend to fall below a particular level of yield. More diverse systems tend to fall above that line. And so here you can see independent contributions of species traits, the size of the organisms, and also biodiversity. So it's not just traits, it's not just biodiversity, it's these two things interacting together in these systems. Now, the final thing that I want to talk about is the discouraging aspect of this talk, I guess, and that is that we know so little about the actual distribution of microbes in nature. And until we have a better handle on this, we're not going to be able to resolve some of the major issues related to microbial biodiversity and ecosystem functioning. The big problem is this. Typically when we go out to nature, there's nature up there, and there's a lake right there. We go to that lake and we take a little tiny sample. And from that we try to extrapolate everything about the biodiversity of that system. If we do a bad job, we simply look at the culturable organisms. If we try to do things better, we extract the DNA from those organisms, and then we use a number of culture-independent approaches to try to understand what's there. The sampling bias involved in going from a large ecosystem to a tiny sample, of course, is a major issue. The other aspect of sampling bias is that all of these culture-independent techniques have their own sampling biases associated with them, and so they tend to undersample or focus on particular groups. And that means that even using very sophisticated approaches like microarrays, community fingerprints, clone libraries, we're still only capturing a tiny fraction of what's there, and that leads to major issues. The other important issue here is that even when we can actually look at the organisms and identify them under the microscope, there are these cryptic species complexes that make us think that there are fewer things there than are actually present. And again, this suggests that we have to use not just these traditional approaches, but molecular approaches to try to understand the full nature of diversity that's there. Until we can get around this problem, we're never going to be able to resolve the issue of whether things are in fact truly cosmopolitan, whether there is extensive biogeography of microbes, and most importantly, how many species fall into the cosmopolitan bin and how many species fall into the bin of organisms that actually display global variation in biodiversity and distribution. That, I think, is the huge remaining problem for not only limnologists and oceanographers to solve, but ecologists working in microbes in all different kinds of environments. And I'll let you think about that for a bit. So I'll just close by thanking the organizers for inviting me and Helmut to come here today and talk about these problems. Wanted to mention that lots of people in my lab have contributed to this work, and a lot of this stuff would not have happened without their effort. And also to thank our various funding sources, the US NSF, NASA, and RUTGERS for making it possible to do these things. So thank you.